Hello viewers, we are from Group 4 Chemical Engineering. Welcome to our video on article critique. In this video, we'll discuss several aspects of the study done on microplastic bioaccumulation in cod fillets from Norway. Let's dive in. With a world so dependent on non-degradable plastic, persistent organic pollutants and microplastics can be found globally in various environments. Marine organisms can ingest these particles and find their way into avians and humans that consume them. This study aims to test the level of microplastic bioaccumulation in cod fillets from plastic-polluted waters and assess the potential risk posed to human consumers.
Next, I will present the methodology and result of the study. The study aimed to determine if cod, a significant species for human consumption, bioaccumulates microplastics. The study involved a sample of 22 cod fish collected from two locations in western Norway during May and November of 2021. For the sample preparation, the researchers dissected 100 grams of cod muscle tissues from each fish. These samples were then cleaned using a gentle enzymatic treatment to ensure the removal of any contaminants without damaging the tissue. While for the detection method, microplastics were detected using microfourier transform infrared spectroscopy, or known as MU-FTIR. This highly sensitive technique can identify particles down to 10 micrometers in size, allowing for precise detection of microplastic within the tissue samples. Next, let's move on to the result overview. The data collected indicated a low concentration of microplastic in cod muscle tissue. The average concentration was 2.81 plus 8.33 micrograms per kilogram of weight, with a frequency of occurrence of 34.78%. Importantly, the study found no significant correlation between microplastic bioaccumulation and the age or body size of the fish. This suggests that cod muscle tissue may not be a primary site for microplastic accumulation, even in areas with high plastic pollution. These results highlight that while microplastics are present, their accumulation in cod muscle tissue is relatively low and does not appear to be influenced by the fish age or size.
Let us look at the strength of the study. Firstly, it was published in the journal Water Emerging Contaminants and Nanoplastics, which is indexed in Scopus, signifying high academic standards. The authors, Mark Huff, Emily Hagerness, and Alessio Gomiero, have strong backgrounds in marine ecotoxicology. This underscores credibility of the research. A key experimental strength is the use of micro-FTIR, which detects microplastic as small as 10 micrometers. This ensures precise and accurate results. Additionally, the study had strict contamination controls in place. The lab used plastic-free surface, such as glass and steel, to minimize the risk of contamination. Moreover, the focus on non-migratory cod, continuously exposed to local pollution, is one of the strengths of the study. This targeted approach provides a reliable indication of microplastic accumulation, making the findings robust and relevant.
Hi, I am Natasha and I will explain the limitation of their recent study on marine pollution. They highlight three critical issues that hamper the study's breadth and depth. Firstly, the sample size was notably small, consisting only 23 cod specimens, which severely constrained the ability to generalize findings across broader population. Secondly, due to regulatory restrictions, younger and smaller cod were excluded from the study, while financial limitation prevented the inclusion of older cod, thus limiting the analysis of age-related microplastic bioaccumulation. Lastly, the absence of a control group from unpolluted waters meant there was no baseline for comparison, impeding a comprehensive understanding of pollution impacts. Despite these challenges, the researchers emphasized that their findings provide valuable insights into marine pollution effects, underscoring the need for larger and more inclusive studies in the future to address this limitation and expand scientific understanding. Next, interpreting pollution data issues. The researchers start by explaining the difficulties they face primarily due to the reliance on indirect pollution data sourced from external study. The most recent data they could access is from a monitoring station located 80 kilometers away and a significant secondary data source back to 2015. This constraint highlights the potential gap between the data relevance and the current environmental conditions. Turning their attention to tissue analysis, the researcher elaborates on the study's narrow focus, which centered exclusively on muscle tissue. While muscle tissue is indeed an important aspect of marine life, studies This focus overlooks critical organs such as the liver and gastrointestinal tract. These organs often accumulating pollutants at different rates and levels compared to muscle tissue. The researcher stresses that by neglecting these organs, the study might miss important insight into how pollutants are distributed within marine organisms and subsequently transferred to human through seafood consumption. The researcher discusses the broader implication of this limitation, emphasizing the need for comprehensive and accurate data. They advocate for improved data collection methods that are more localized and current, like broader tissue analysis that include various organs. By doing so, future study could provide a more holistic view of pollution dynamics. As they conclude, the researchers underscore the importance of overcoming these methodological challenges. They argue that only by addressing this issue can the scientific community offer more precise insight into the dynamic pollution.
Moving on to recommendations for future research. First, it is crucial to increase the sample size and include a broader age range of koi. This would provide a more comprehensive understanding of microplastic bioaccumulation across different life stages. Second, incorporating a control group from pollution-free water is essential. This will allow for accurate comparisons and validations of the study's findings. Third, directly measuring microplastic concentration in the specific sampling locations would improve the accuracy of pollution data. This ensures that the findings truly reflect the environmental conditions where the cod inhabit. Lastly, analyzing additional tissues, such as the liver and gastrointestinal tract, is important. This will provide a more holistic view of microplastic presence throughout the fish body, offering better insights into potential risks for human consumers. Implementing these recommendations would enhance the reliability and comprehensiveness of future studies on microplastic bioaccumulation in marine life.
Now, let's conclude with the main points from our critique of the study. The essence of microplastic bioaccumulation in cod fillets from plastic pollution in western Norwegian waters. The study found minimal microplastic bioaccumulation in cod fillets, suggesting a low risk to human consumers. However, these findings should be interpreted with caution due to several methodological limitations. This research highlights the need for more comprehensive studies to understand the impacts of microplastic on marine life and human health. As plastic pollution continues to rise, knowing how it affects our food is crucial for public health and environmental policies. Future studies should address current limitations such as increasing sample sizes, including diverse age groups, and directly measuring microplastic concentrations in the fish habitat. This will improve the reliability and understanding of the risks associated with microplastic pollution. Thank you.